welcome to the July 2017 Nutritionist webinar. I am Marianne Fessenden from AMTS and your English language host. These webinars provide access to technical seminars by internationally recognized speakers. This series presentations are held in English, Portuguese, and Spanish. Paula Torillo from Argentina is hosting the Spanish language webinar. Marcos Neves Pera from Universidade Federal de Lavras will be hosting the Portuguese language presentation with Marcelo Hens Ramos from 3R Lab facilitating. There will be a question and answer period immediately following the presentation. Listeners can submit questions through me, Marcos, or Paula. A complete recording of archived webinars as well as the question and answer session for each will be available on the AMTS website. We are very pleased and honored to host Dr. Dale Bauman, a professor emeritus at Cornell University. Dale Bauman is Liberty Hyde Bailey Professor Emeritus in the Department of Animal Science and the Division of Nutritional Scientists at Cornell University. Raised on a dairy farm in Michigan, his undergraduate and graduate degrees were from Michigan State University and the University of Illinois. Bauman's research efforts involve an integration of nutritional, biochemical, and endocrine aspects of metabolic regulation with an overall goal to 1. Provide a conceptual basis broadly applicable to all physiological states in biology, 2. Form the biological basis to enhance the efficiency of animal production, and 3. Provide an opportunity to design animal source foods consistent with consumer perceptions and health recommendations. With over 800 co-authored scientific articles and 32,000 scientific citations of his research, Bauman is among ISI's highly cited scientists and Science Watch's list of most influential scientific minds. A recipient of the Merrill Outstanding Undergraduate Educator Award from Cornell University, 35 of his graduate students and postdocs have gone on to become faculty members at universities around the world. Elected to the National Academy of Scientists in 1988, Bauman received the USDA Distinguished Service Award and the Alexander von Humboldt Award for U.S. Agricultural Research. Bauman has been a member of several NAS and USDA advisory committees and served as chairman of the NAS NRC Board on Agricultural and Natural Resources and as president of the American Society of Nutrition. He has received awards from various scientific and professional societies, including ADSA, ASAS, ASN, Council of Agricultural Science and Technology, Federation of Animal Science Societies, and National Dairy Shrine. In addition, Bauman has received alumni awards from Michigan State University and the University of Illinois, Career Achievement Award from Cal's Cornell University, and an honorary doctorate from Michigan State University. Dale was recently the subject of an ADSA symposium honoring the contributions he made to industry, and that was quite a long introduction, but he's pretty neat. I should also say he was one of my most inspiring professors when I was an undergrad and took his senior seminar class. Dale, welcome to the Nutritionist. I'm going to unmute your mic, so if you want to test and say hello.
Hello, that was a long introduction.
It was. I pulled it from you, but it was worth it. Now I'm making you the presenter, so you'll be able to progress through the slides.
Okay, let's get started. So today, this evening, I'm going to talk about dietary-induced milk fat depression, and both provide a background in terms of fatty acids and lipid metabolism a bit, then talk about historically what has gone on in this area, and then most recently where we've been able to figure out exactly what the cause of this is. Let's start here. This is a beautiful tracing of an electron micrograph that Bill Held did. And we have here 3 mammary epithelial cells. Active cells, this is how they would look in a cow's mammary gland. Down here at the bottom on the right is the myoepithelial cell that will respond to the oxytocin for milk letdown. And on the left, this is a capillary with a red blood cell. So we're going to be talking about milk fat synthesis today. And so this clear globule is milk fat. It was made in the endoplasmic reticulum and then coalesced into this fat droplet. And you can tell here on the left, when it's secreted into the milk fluid or the lumen of the mammary gland, it captures a membrane from the cells. So the lipid droplet is encased in a fat globular membrane. And the mammary gland is really an amazing biological factor. In fact, an article in Scientific American suggested that the mammary gland was second only to photosynthesis in terms of biological reactions. And in terms of milk fat, it's really a lipid synthesizing machine. Just amazing the amount of lipid that's made. Let's consider the importance of milk fat and some of the properties that make it of good value. Number one we would list would be the flavor, texture, and manufacturing properties of products. And to a very large extent, the flavor of dairy products is really related to the milk fat composition. As you're aware, milk fat production also requires a considerable portion of the energy requirement for the cow. And we have a situation most recently where we have an increasing consumer demand for milk fat. In particular, the consumption of butter and cheese has increased dramatically, even though the fluid milk has gone down. And that's partly because there's been a realization in the last few years that the old ideas that saturated fat represented a health risk were simply incorrect. And gradually the dietitians and the nutrition recommendations for consumers have begun to be modified to reflect the fact that the milk is a highly desirable, milk and dairy products are highly desirable for their nutrient content, and that the saturated fats in milk do not represent a health problem. So if we look next at some of the characteristics of milk fat, and we'll spend the first few minutes of this lecture talking about the milk fatty acids. I mentioned already and showed you the picture, the micrograph of the fat as a lipid droplet surrounded by the phospholipid membrane called the milk fat globular membranes and proteins. The fatty acid, the fat in this globule is mostly triglycerides. That's a glycerol backbone with 3 fatty acids esterified to it. And in general, we can characterize the milk fatty acids into 3 categories. And this becomes important for us to understand the dynamics of milk fat production. The first is what we call de novo or newly synthesized fatty acids. And these would be fatty acids that are shorter than 16 carbons. The second, or let's go up down here, the second would be what we call preformed fatty acids. These would be fatty acids. The de novo are synthesized in the mammary gland newly. The preformed are fatty acids taken up from circulation. So they are derived either from absorption from the digestive tract or mobilization of body fat stores. And these are the carbons, the fatty acids longer than 16 carbons. And then in the middle we have a mixed source, that's the 16 carbon fatty acids, and they come from both sources. And so roughly 1/2 of the 16 carbon come from de novo and 1/2 from preformed. Now, milk fat is really diverse in terms of its fatty acid composition. There are over 400 different fatty acids in milk fat, and this includes many bioactive fatty acids such as the CLA isomers and branched chain fatty acids. And part of the reason, a large part of the reason for this large number is due to the rumen fermentation and the fatty acid byproducts made in the rumen. The structure of the fatty acids then, we think of ruminant products as being saturated in fat. And so we have here a 4-carbon chain. The carbon has 4 bonds, so 2 of them are occupied with the adjacent carbons, and then 2 of them have hydrogen. And so that's a saturated fat. Unsaturated fat then loses 2 of the hydrogens, and instead you have the bond between these 2 carbons. And so ruminants are considered saturated, having saturated fat because of the rumen microbes converting unsaturated fatty acids to saturated. In reality, though, only about half of the fat, or even less, in ruminants is really saturated. Of the unsaturated fatty acid structures, the double bonds can take 2 forms. One is a cis form, and that's shown in this diagram, and that's a situation where the 2 hydrogens are on the same side of the molecule. The other form is a trans form, and that's a situation where the 2 hydrogens are on opposite sides of the molecule. Now, this makes a big difference in terms of the structure of the molecule, because trans fatty acids are more a straight chain, and the cis double bond causes kind of a dog leg shape to the fatty acids. Trans fatty acids, particularly the trans fatty acids arising from partial hydrogenation in preparing some of the oils, of course, have received a lot of publicity because they're associated with cardiovascular disease. That's a little different than the trans fatty acids, though, that are in dairy products. Finally, I have to show you one more structure. The double bonds then can either be conjugated or non-conjugated. Normally in nature, they're non-conjugated, meaning there is an interceding carbon between the double bonds. So you have a double bond, then a carbon, and a double bond. And these 2 would be, as drawn, a cis structure. Shown down here then is a conjugated double bond. Conjugated meaning there is no interceding double bond there is no interceding carbon between the double bonds. And that again gives it very different physical properties, but as we will learn as we progress here, it makes a tremendous difference in the biological properties of the fatty acid. Conjugated double bonds then, or the CLAs, conjugated linoleic acid, is a common form. And this comes about because of the rumen microbes, and this is an intermediate in the biohydrogenation of unsaturated fatty acids in the rumen. So there we've got a bit of a background on the structure. Let's look at a typical composition of dairy milk fat, and that's shown here. These are survey data over a full year period in the United States, so they're average data across all geographic regions and seasons. And if you counted, you would see here there are bar graphs listed for 13 different fatty acids. Even though there are 400 fatty acids in milk fat, these 13 are the major ones, and even they, you see, differ. Starting on the left here, we see C4 through to beyond 14. These would be de novo synthesized fatty acids. And you can see they're all have the carbon length and then the number of double bonds. So on the left side of this diagram, they're all saturated, no double bonds. Here's palmitate, C16, stearate, 18, and then we begin to have the unsaturated fatty acids. The major one being oleic acid, C18-1 cis. I mentioned about conjugated linoleic acid, and here's conjugated linoleic acid. You can see it's very small. The trans acids are very small. The omega-3 fatty acids are relatively minor. These other 385 different fatty acids that are in milk fat then are represented here. So these 13 are the major ones that are in milk fat, and we'll be talking a bit about CLA and the trans fatty acids as we go along. So, as you're aware, milk fat is affected by many factors. A portion of those factors are non-nutritional, and they involve genetics. For example, the colored breeds typically have much higher milk fat than do Holsteins. Stage of lactation, milk fat being higher in early lactation and later lactation. Season affects it. Parity and temperature all have an impact on the amount of the percent milk fat in milk. The nutritional factors are listed on the left, and these include things such as fiber in the diet and certain types of feeds, for example, oils or fat supplements, different feeding strategies, frequency of feeding, for example, and ionophores. And so milk fat is among the most variable then of milk components. The lactose will be relatively constant, always about 5%, because it's the osmoregulator of milk volume, so it's always 5%. Protein also tends to be constant unless you have inadequate supply of amino acids. But milk fat can vary quite widely due to these various factors. Well, one of the situations then that was recognized over 150 years ago was what we call the low-fat milk syndrome, or diet-induced milk fat depression. And Buzingay was the first, a French scientist was the first one to recognize this and comment on the fact that diets seem to cause a variation in the milk fat content. Buzingay is an interesting man. He's considered the father of scientific research in agriculture. So there are certain diets that this occurs with. Low fiber, high grain diets, diets that are high in oils, fish oil, for example, would all be diets where you typically would see milk fat depression. And this is unique for milk fat. The milk fat can be reduced up to 50%, but other milk components are not affected. So the actual production of the cow, the content of lactose and protein are not changed, but milk fat can be reduced by 50% or thereabouts. And in particular, it became a problem, a recognized problem, in the 1940s when we began to use scientific feeding. When we had the first NRC bulletin for dairy cows came out in the early 1940s, and they began to feed cows according to recommendations, and that meant types of diets that are more associated with milk fat depression. In particular, it was recognized in 1939 at the World's Fair. This kind of represents a landmark situation because the clear manifestation of the problem occurred in this Borden's Dairy of Tomorrow, an exhibit that they put on. So they had a rotary parlor set up here with 10 cows from each of 5 breeds, and a situation so the public could view these. And so these cows were confined in this unit, and they were fed a pelleted diet. And so that caused quite a dramatic reduction in milk fat, a reduction enough that they had trouble making butter to show the consumers what butter looked like. So that was clearly a situation that caught people's attention. So there began then an effort that lasted over the rest of that next 50 years to try to unravel the cause of this milk fat depression, because as we began to feed commercial diets, it was a problem very often. And so the theories as to what caused it were divided into two broad categories. One was a shortage of precursors to make the milk fat, and the second was there's some type of inhibition of milk fat synthesis. The precursors, this has to do in particular with the fatty acids synthesized de novo, because these were fatty acids that were most dramatically affected in milk fat depression. And one theory was that you had a decrease in the acetate production, because acetate furnished most of the carbon to make the de novo synthesis of milk fatty acids. And of course, that was coming from rumen fermentation. The second theory was the beta hydroxybutyrate deficiency, another product of rumen fermentation. Beta hydroxybutyrate furnishes the first 4 carbons of the de novo synthesized fatty acids. So if that was inadequate supply, we would have a problem. And then the final theory here was a glucogenic insulin theory that I'll explain in a moment. So if we look first at the phenotype you would typically see, so here's an example related to the actual acetate shortage theory and also the beta hydroxybutyrate shortage theory. So we have 2 columns of data then, a normal diet, and then cows fed a high grain, low fiber diet. So here's the milk fat produced. These cows are identical in milk production, but have about a 45% reduction in milk fat synthesis, milk fat output. And one of the things that was observed when you saw the high grain, low fiber diet that caused milk fat depression was you had a lower acetate to propionate ratio. Whereas the ratio might normally be 3.2 to 1, now it had decreased to 1. So that gave rise to the idea that you would have an acetate deficiency. However, as the science moved forward, we were able to get radioisotopes to study the actual production of acetate in the rumen and quantify how much was made. And you can see the rumen production of acetate in moles per day in this row. And you see there really was no difference in the acetate production between these 2 diets. And rather, this marked difference in the acetate to propionate ratio was due to the high propionate production that occurred on these high grain, low fiber diets. So these type of diets cause a lower rumen pH, change the microbial population, and the population that's there produces a higher amount of propionate. And in the case of beta hydroxybutyrate, you see also that here's the production rates, and there really is no difference there, suggesting that you had a deficiency of beta hydroxybutyrate. So there really was no support for the idea that you had a shortage of acetate and beta hydroxybutyrate. However, the high propionate then is really the basis for this next theory, the glucogenic insulin theory of milk fat depression. I should mention the VFA the measurements of acetate and propionate and butyrate production, that was done in the 60s, 1960s, by groups like Carl Davis and Dick Brown at Illinois. This theory then was the idea that when you did feed a high-grain forage diet, you would increase the propionate production. Propionate is absorbed and converted to glucose in the liver, and glucose is a secretagogue, a stimulant for pancreatic production of insulin, so you'd have more insulin, and that this in turn would cause milk fat depression. So the basis for this is shown here, where insulin turns out that it has absolutely no effect on the uptake of nutrients and the rates of fat synthesis in the mammary gland. But insulin does stimulate adipose tissue rates of lipid synthesis. In particular, it stimulates the uptake of the precursors. And so the idea was if you had higher propionate, you would have higher insulin, and this really would cause adipose tissue to take up the nutrients. The mammary gland would have short supply and therefore not be able to synthesize the quantity of milk fat needed. So I often refer to this as a competition theory, a competition between adipose and mammary, affected by insulin, in which adipose was winning the competition. That's a good idea in the sense that a lot of the pieces seem to fit with that, but in a dairy cow, mammary milk synthesis has a really high priority. And the idea that adipose tissue would somehow compete with mammary gland and win a competition just didn't seem very likely. However, it was a theory that held through the late 70s, 80s into the early 90s. And many of the applied things people tried to do then was based around this theory. However, infusions of starch, glucose, or propionate, many studies, really didn't consistently show an effect on milk fat synthesis. The other thing was the changes in milk composition you saw when you did see a milk fat effect were different than what occurs with diet-induced milk fat depression. And so overall then, the studies really provided little or no support for theories involving a shortage of precursors for milk fat synthesis. The second type of possible cause of the milk fat depression then was the idea there was a direct inhibition of milk fat synthesis. And that was an idea that actually went back to some comments that Carl Davis and Dick Brown had included in a review. And a particular key study was one done by Mikko Greenery, in which he designed diets that were able to separate that you really needed two conditions to get the milk fat depression. You needed an altered rumen fermentation, which had been recognized for some time, because the altered rumen fermentation was associated with changes in the VFA molar ratios. However, the part that was really unique was he designed diets that had very little, negligible amounts of polyunsaturated fatty acids. And when that was done, then you really don't get milk fat depression unless you have both of these conditions. You can alter the rumen fermentation, but if you don't have polyunsaturated fatty acids, you will not get milk fat depression, and vice versa. So these two conditions are really telling us something particularly significant. They're telling us that this really has to be an interaction between processes occurring in the rumen digestive tract to be associated with rumen fermentation. And it has to also then involve mammary synthesis of milk fat. Now, this makes the problem challenging to study. You really can't put it in a test tube or a culture system or an in vitro system because you need both the rumen digestive processes and the mammary synthesis of milk fat to look at this problem. So you really have to do the work in vivo. And the idea was, or one of the early ideas, by now we're into the 1990s and in the first part of this century, the trance theory of milk fat depression, that when you have this shift in rumen fermentation, you alter the rumen microbes, so you end up forming trans fatty acids in the rumen, some unique trans fatty acid isomers in the biohydrogenation process, and some of these must get absorbed and inhibit mammary fat synthesis, causing milk fat depression. So this theory sounded good. Our challenge was we really couldn't analyze these different intermediates of fatty acids. This captures it in terms of the cow. So the cow consumes a diet. The diets, our typical dairy diet has unsaturated fatty acids in it. The rumen normally biohydrogenates almost all of these fatty acids. But in this situation, the idea was you had some intermediates that were escaping rumen fermentation that were adversely affecting rates of milk fat synthesis. Look at rumen biohydrogenation then in kind of a simplified style. Shown here are two of the major polyunsaturated fatty acids in ruminant diets. Linoleic acid, C18-2, and linolenic acid, C18-3. And you can see the progression is to, let's look at linoleic first. To first change this double bond, move this double bond, so you have cis-9 trans 11. And in turn, then you hydrogenate the cis-9 double bond to give you trans 18-1, trans 11. And then a second hydrogenation step to give you stearic acid. And linolenic acid has a similar progression, a little more complicated if you draw in all the possibilities, but it still goes through trans 11, 18-1. And these two fatty acids, cis-9 trans 11 CLA, conjugated linoleic acid, is given the common name rumenic acid because ruminant fats are where you typically would find this. And trans 11, 18-1 is given the common name vicinic acid, milk fatty acid, in terms of its translation, because it's the major trans fatty acid in milk fat, trans 18-1. Now, the theory, or our theory, was that there were some different intermediates made here. And so we could not separate all the different trans 18-1 isomers. And so again, Mikko Greenery really worked out through a sequence of different runs with the gas chromatograph to separate out the different isomers. And so here's the profile of trans 18-1 isomers in normal milk fat. And the major one is vicinic acid, trans 11. And you can see when we were finally able to separate these, lo and behold, trans 10 18-1 increases in milk fat depression, suggesting that must be involved in some pathway that becomes important in this process. So if we draw the schematic, instead of going this typical way to make the rumenic acid and vicinic acid and then stearate, it must be going off to alternative routes to form this trans 10 18-1, and that would involve this intermediate trans 10 cis 12. When you look at linoleic acid, it means instead of first moving the cis 12 double bond like you have here. Instead, you move the cis-9 double bond. And so you have trans-10 cis-12 CLA. So something about this seemed likely to be related to milk fat depression. Now, none of these unique fatty acids are available commercially that you could buy them, even small amounts for standards. So that provided lots of challenges. And we had to work in vivo. So the way we were able to do this was to abomasally infuse some of these fatty acid isomers for a few days and look at their effect on milk fat. And by going through the rumen fistula with this setup here, we could in fact infuse into this bottle that was in the abomasum. This collar kept the bottle in the abomasum. And we could get a chance to bypass the rumen, so we didn't have the bugs altering the fatty acids, and figure out what they were doing. And so here's the very first study done by Lance Baumgart, where we compared these two CLA isomers, rumenic acid and then the trans-10 cis-12. And you can see we did a 4-day infusion. Once you start infusing at 10-12, milk fat immediately dropped in half. We stop infusing, it comes back. Now we're infusing very, very small amounts. So this fatty acid is really bioactive. Together the theory, the biohydrogenation theory of milk fat depression, that under certain conditions, rumen biohydrogenation results in unique fatty acids that are potent inhibitors of milk fat synthesis. For example, trans-10 cis-12 CLA, and possibly other related intermediates from the polyunsaturated fatty acids. So with this, then, we had a cause, and we also had a direction we could go in terms of figuring out applied situations and practices that could minimize that. Now let me show you this gas chromatograph of milk fatty acids just to show you the challenge we were facing. Because this all happened about 10, 15 years ago. So here's the control fatty acid profile, and here's the milk fat depressed. Can you see the peaks that are different? Well, you can't see them because they're right here in this region. The next slide, I'll expand that region. And here's that region expanded. Here's a major CLA isomer in milk fat, rumenic acid, which doesn't have an effect on milk fat synthesis. And here's trans-10 cis-12. So you now have perhaps a better feel for the challenge it was to identify this, but also what very small amounts of these bioactive fatty acids are really potent. Dose response curve infusing different amounts of trans-10 cis-12. You can see how it goes down very fast and gives you your typical dose response curve. It turns out that this inhibits milk fat synthesis in most all species, including humans. But an interesting thing, when we compare what the curve looks like with diet-induced milk fat depression, you can see this is a little steeper curve for the level of 10-12 versus milk fat depression. And so one explanation for this is there would be other fatty acids that are causing the depression. So we went after that. It means that we really are altering the pathways, and so other isomers could be involved. And so as we got into this, it turned out to be a much more complicated situation than we fully appreciated. Here's rumenic acid. In milk fat, it's 73 to 91% of the CLAs. But look at all these other fatty acids, CLAs, that are in milk fat. Here's 10-12. And so knowing this level, these small levels can have an impact, any of these could as well. We began to try to sort that out and identify which ones would be involved. To do that, we really had to synthesize some of these ourselves. We worked with Natural in Norway, Asger Sabo to get some of these. We worked with Nestle Nutrition in Switzerland. We worked with FDA Food Safety Division to synthesize some of these, and we had quite a number of these isomers synthesized so we could test. Here's the ones we've tested that cause milk fat depression. So at this point, we've really identified 3 that seem to be associated with milk fat depression: trans 10 cis 12, cis 10 trans 12, and the reverse isomer for rutenic acid, trans 9 cis 11. My guess would be there are more, but it's difficult to study them. We and others have also looked at some of the trans 18-1 isomers. And we're able to infuse larger amounts of these because they're present more. And with some inconsistencies, it seems like all of these are incorporated into milk fat, and none of them affected milk fat yield in any substantial manner. 10 is the interesting one, though, because it turns out this plot shows the relationship between the milk fat content of trans 12 and milk fat percent. Trans content of milk fat and milk fat percent. And you can see how the more trans 10 there is, the lower the milk fat, and quite a sharp decline. Some of these are diets even you don't think of having milk fat depression, but it seems like that on most diets even you have some small degree. And so trans 10, while it may not be causing milk fat depression, it is definitely a great marker for this altered rumen fermentation associated with milk fat depression. So you can, and it's much easier to analyze than the CLA isomers. So you can analyze this in your milk fat and tell if this is the direction you need to go in resolving your problem. Now then, we have a situation that milk fat is, this specific fatty acids, bioactive fatty acids, are somehow shutting down milk fat synthesis. How are they doing that? And so that really has to involve gene expression. And so this is the regulatory arm of the gene. We have shown here the promoter and activator repressors. And so some transcription factor or activator repressors coming in and modulating the expression of some of the key genes for the enzymes involved in milk fat synthesis. And so we considered that. Here are the different proteins, fatty acid sensor proteins, that is, proteins that are known to regulate some of the fatty acid synthesizing genes. And in particular, these over here, these 2 are key ones, SREBP and S14. And so initially worked by Dan Peterson, but in particular then Kevin Harvatine did classic studies identifying these 2. And so you have a situation shown here. In the right-hand profile is a milk fat percent, so we have control orange. Aqua is the CLA isomer when we're infusing 1012, and blue is when we're feeding a low fiber, high oil diet. So you see how milk fat goes down. Here's how the level of this regulatory protein, SRABP, goes down the same way. And so the expression of SRABP is highly correlated. And in particular, these key genes involved in lipid synthesis have a region in their gene that recognizes the SRABP, active complex of the SRABP, and shuts down the expression of the gene. And so key enzymes such as carboxylase, synthetase, and so forth, all are coordinately downregulated when you have milk fat depression. They all contain this promoter region for this active form of the SRABP, so they're all regulated by SRABP. So that's been an amazing story, bringing that all together. I'd like to digress just a moment before I wrap it up here to make the point that we're dealing here with bioactive fatty acids. So here's cis-9 trans-11 rumenic acid, and here's trans-10 cis-12. So this one causes, here's the double bonds. This one causes milk fat depression, and in fact it inhibits lipid synthesis in lots of tissues. This one does not. The double bonds are just moved one location. However, this rumenic acid is a potent anti-carcinogen and also anti-atherogenic. So this rumenic acid is the one, the CLA you hear about, that we're trying to increase the level in milk because it's a fatty acid that has functional food properties. And the concept is that this is part of this bioactive fatty acids that are allowing, that are helping in health maintenance and the prevention of chronic diseases. So it's amazing, just moving that double bond has this dramatic effect in the biological role that these different fatty acids play. So we have a lot to learn. In humans, they've called this the microbiome era, and so you hear about different microbiomes having a role in preventing chronic diseases. So let me summarize then. Milk fatty acids, milk fat depression is caused by bioactive fatty acids that originate from rumen biohydrogenation of unsaturated fatty acids. So it's from the partial biohydrogenation. And the rumen environment is critical in this. It really involves interactions on quite a number of factors. These bioactive fatty acids then, if they pass to the small intestine, they're taken up by the mammary gland, and they act to downregulate expression of the genes for key enzymes that are controlling milk fat synthesis. And finally, I tried to make the point that not all fatty acids are created equal, and the location and geometric configuration of the double bonds results in marked differences in the biological effects of these fatty acids. The term nutrigenomics, the idea that the dietary components or components absorbed from the digestive tract, in addition, they can provide nutrition, but they also can have effects on regulation of gene expression. Gene expression, epigenetic effects. And so this is a whole new era that's being looked at. And diet-induced milk fat depression really represents nutrigenomics in action. And of course, it's really biologically significant. Finally, I wanted to list some of the people that have contributed to various parts of this, both in our group and interactions, but also people, FDA and Nestle and Natural, that were involved in making intermediate work. And with that, I'll try to take any questions you might have.
Okay, Dale, I'm going to do I'm going to do a couple housekeeping details and introduce the next webinar presenter, and then we'll go to questions. If the audience wants to write any questions they might have in the question and answer window, I will ask them for them. So first, let me introduce our next month's webinar. Our next month will feature Dr. Christian Kruiwagen, an emeritus professor in the Department of Animal Sciences at the Stellenbosch University of South Africa. After working in the Department of Agriculture as a research scientist for 21 years, he joined the Stellenbosch University in 1996. His research focuses on the nutrition of dairy cattle and calves, and he has 116 scientific and popular articles to his credit. Dr. Kruiwagen is a member and past president of South African Society of Animal Science and a member of the American Dairy Science Association. He is also the former chair of the Agricultural Microscopy Division of the American Oil Chemists Society. Awards include the South African Society for Animal Science Bronze Medal, the Animal Feed Manufacturers Association Technical Person of the Year in 2009, Western Cape Province's Agriculturist of the Year in 2009, and the Stellenbosch University Rector's Award for Research Excellence in 2010. In deference to Christian, who is 6 hours ahead of us, there will be us here in New York, and in consideration to audiences for whom the 6 p.m. time slot presents a challenge, we will hold the webinar next month at 1 p.m., and that will be August 9th, 1 p.m. I also want to thank our partners and sponsors, AMTS, USA and Global, 3R Lab, and Paula Turillo. The gold sponsor is Ajinomoto Heartland. The silver sponsors are Arm & Hammer Animal Nutrition, R&D Life Scientist Virtus makers of Stratus with EPA and DHA omega-3s, and Prequel with omega-6s, Dairy One Forage Laboratory, and Dairyland Laboratories. Our bronze sponsors are Jeffo, Life Made Easier, ADSA, AminoMax, and Quality Liquid Feeds. I'm now going to open up the floor for questions, so if you want to go ahead and type it in, I'm going to allow our Brazilian hosts to ask their questions first because they actually start the webinar in Portuguese after this one's finished. So I'm going to open up Dr. Bauman's microphone and also Marcos Pierre. So Marcos, if you have questions, go ahead. And Dale, you're unmuted, and I'm giving the presentation back to you so that you can move slides.
Okay, thank you. Thanks, Dr. Bauman, for the talks. Very clear, very direct. And I have a question here. Someone wants you to comment on the use of the milk fatty acid profile on troubleshooting nutrition in dairy herds, like probably using the preformed or mixed or the de novo or the trans 10 to trans 11 ratio, or what do you think the potential, how it can help the nutritionist?
So the idea is, the question really centers around some of the work initially spearheaded by Dave Barbano, and depended on being able to analyze the milk fatty acid profile more quickly, at least in broad categories. And so that is currently being tested at the recent ADSA meetings. There were quite a number of presentations related to that. I think the potential is good. At this point, though, to me, it still hasn't quite evolved to the place where we can confidently predict or know exactly how to use it. It is clear, as you could probably tell from my presentation, that under some of these situations, you definitely do see a change in the fatty acid pattern, and the extent to which the different situations match up with that profile is what's being characterized now. You really are interested in using that for a lot different situations than milk fat depression. You're interested in using it as a fine-tuning management technique, and at this point, I think it's still in the testing stage, but it has really interesting potential.
Okay, thank you. I have another question here. Assuming that the cow is producing the same amount of milk, like milking frequency, like 2 times a day, 3 times, or 4 times, would that affect milk fat concentration at a similar milk yield, I assume?
Well, I'm trying to recall. When you increase the milking frequency, you increase the milk yield, and you also increase the milk fat yield. So I think that's a situation where the yield of the major milk components would move together: lactose, fat, and protein. There are other situations where they don't necessarily move together, but in that case, I think they would.
Okay, have another one, the last one. Sometimes we get the response in milk fatty acid profile as a percentage of the total fatty acids. And sometimes we get the response in secretion per day. Like, how would you interpret that? Like, for example, the secretion of CLAs in grams per day, the kind of what the mammary gland is making, and we talk a lot about the proportion of the VF of the fatty acid, but sometimes we don't get anything on the proportion, and we get on the secretion.
You get it on quantity. Yeah. That's a good question, and that's part of the reason for really understanding the different fatty acid composition and what that exactly means. You have different situations where sometimes people will contact me and say they have milk fat depression, and when I look at their situation closely, they don't have milk fat depression. They just have one of these situations where milk yield has been affected, and in turn you end up affecting fat yield. So you need to consider that in evaluating the situation. I think in terms of what I'm talking about, you see a change in the quantity, and when you look at the pattern, then you will see a change you'll see a difference in the pattern. If the pattern isn't changed, then I think quantity differences, you're really not dealing with a diet-induced milk fat depression per se. You're dealing with some other physiological situation. You also will sometimes see, when you're looking at the pattern, you will see the long chain fatty acids increase, and you will see in the quantity. You will not see the short and medium chain fatty acids decrease, but you'll see the long chain fatty acids increase. So that's also a little different situation, and that almost always is a situation where your animal is mobilizing body fat, and the fat that's mobilized is taken up quite extensively and incorporated into milk fat. And so anytime you have a situation where you increase fat mobilization from body reserves, you will see milk fat percent go up, and that increase in percent will be due to longer chain fatty acids.
Another one here. What would be your safe limit for unsaturated fatty acids in the diet?
Yeah, that's a good question. I don't tend to look at it that way because it's so variable. You obviously want to ask the question, what is your unsaturated fatty acid load? What fruitful? even when you're looking at unsaturated fatty acids, of those, the C18 one will no fat depression. But it depends on the source, the different sources. It depends on the forms those are in. For example, if they're salts, if they're calcium salts, for example, those are more slowly released in the rumen. They're not inert, but they're released. They dissociate slow enough in the rumen. They tend not to produce some of the biohydrogenation intermediates. It depends on how you feed them. If you're feeding more frequently, that's less likely to have a complication. If you're slug feeding, and so they're getting a larger dose of the polyunsaturates, particularly 18-2 at one time, that's more of a problem. And so you have to consider a number of different things. In the U.S., oftentimes the milk fat depression ended up being associated with distillers' grains, where the fatty acid composition, the percent fat, and the type of fatty acid varied in ways it would cause eating C16 or saturated fatty acid supplements, then those don't cause the problem, and in fact those are palmitate. Dietary palmitate is transferred to milk fat with about 15 to 20% efficiency. So 15 to 20% of the dietary palmitate will be taken up by the mammary gland and incorporated in the milk fat. So the answer to the question is complicated, and it really depends on the concentration of fatty acids in the basal diet, the diet type, and a lot of conditions related to how you're feeding the animal and that rumen fermentation, changes in the rumen fermentation.
Oh, thanks a lot for the talk. I'll go on to the other one.
All
Thank
right.
you.
Bye,
Have a
Marcos.
good night.
You
Bye-bye.
too. Bye. Okay, so we have a number of questions. I know that Paula in Argentina has probably many questions. I've had some questions coming in, and what we'll do is ask a couple questions from the English speaking audience, and then Paula will ask a few questions from hers. So if you have been writing in questions in the chat or the question and answer window, I do see them, and I will get to them. I'm going to ask a couple from Tim Davies in the UK, because I know that he's probably struggling to keep his eyes awake. He says I know he had a cup of tea about 3 hours ago. He had a question that he sent me 3 hours ago, Dale, and he says, I'm just reading a paper by Adam Locke about RUFLs, rumen unsaturated fatty acid load, and he thinks that calculating RUFL would be potential indicator for milk fat depression. How do you calculate RUFL? Is it simply calculating the grams of oleic, linoleic, and lineoleic provided by the diet, or would you put a different weight on the different fatty acids? And I recognize that you may be answering some questions repeatedly, but probably they are worthy of that. So go ahead with that one.
So I'm not so good at questions like that, because my background is more the nutritional biochemistry. But the RUFL, when you calculate the rumen load, I'd have to look up to see exactly how they calculate that, but you're just summing the rumen load. But I know the linoleic acid, the 18-2, is the one that's most important in that load when you're looking at that. And so these are all intended to be guidelines to help you in avoiding or in troubleshooting the milk fat depression situation.
Okay, and I think I was hoping Adam would join us, but I think he's on holiday.
How about Tom Jenkins? Is he on?
I don't see Tom on my list.
Jenkins is the one who started the concept or recognized the concept of RUFL.
Yep.
And it's just logical that the total load of unsaturated fatty acids is really going to be an important consideration in the dynamics of the rumen biohydrogenation.
Okay. And well, I would advise our listeners to look at the archives because Tom Jenkins was on a year ago May, and Adam is lined up for 2018. So I know that milk fat depression is a big concern to a lot of producers, and we're trying to sort of set up the webinars so that we can accommodate that.
I think it's a fast-moving field too. Now that we've identified the cause, we can really zero down and work better to consider how the interactions occur in some of these risk factors. And perhaps even by that time, there will be more information on how to use the milk fatty acid, the overall milk fatty acid composition for management decisions.
I did have a comment from Jim Aldrich, who's listening, and he said RUFL is a sum of 18.1, 18.2, and 18.3, but 18.2 should be weighted, as you said.
Yeah, 18.1, 18.2, and 18.3 are really the unsaturated fatty acids that are in the diet. So
okay,
they're the major ones.
Sorry.
Yeah, good point.
A second
Thanks,
question
Jim.
I had from Tim is how would you go about calculating the acid load of a diet? Would it, for example, be a case of calculating the grams of lactic acid supplied by individual ingredients, and would that tell us anything?
I am sorry, but I can't really answer that. Yeah.
You know what, and a lot of these questions, when we get to something like that, sometimes I fire them off to the various people and have them look at it, and then they get back to them. So I will make a note. And then I'm going to do one last question.
So lactic acid, you're dealing really with the rumen fermentation,
Mm-hmm.
and the idea that when you have a lower rumen fermentation, you're into the environment that promotes those types of bacteria that would be making some of these unusual biohydrogenation intermediates.
Okay. And then a last question or comment from Tim Davies is let's see, what are the key drivers for the switch towards synthesis of trans 10 and cis 12 CLAs
versus normal biohydrogenation?
Yes.
Well, certainly I can make a list of some of the associated effects. Certainly the dietary fatty acids, the type, the profile, and the level of them is going to make a difference. How available these fatty acids in the diet or in a supplement are. If they're less available or they're more gradually become available, then they don't over-challenge the biohydrogenation system. The dietary carbohydrate profile and the rate and extent of fermentation, those would all be part of the story because that'll change the type of rumen bacteria, microbiota that are there to affect the biohydrogenation. So that's why things like effective fiber and the dietary carbohydrate profile come in. Things like feeding strategies in management too, because frequent feeding, ways to avoid a high load of the polyunsaturates into the rumen. So frequent feeding, avoid slug feeding, frequent pushback of feed, all those would be things you could do that would tend to reduce your risk. Types of forage makes a difference because of the forage effects on the fermentation. So those would be a few things that are major.
I do have a comment from, and I'm going to say this last name wrong, but you know who it is, Miko Greenery.
Yeah.
Greenery. She?
So he's in
He.
Finland.
Okay.
Think of him getting up.
Well, he said thanks for a really nice summary, and he's going to sign out and go to bed. So, and it maybe he's already off.
Paul, how nice that he's on.
Yes. I lots of people were very interested. Paula, I'm going to see if you can talk. Are you good now, Paula?
Hello, I'm here. Can you hear me?
Good. Yes.
Perfect. Okay. Thank you very much, Dr. Bauman. A lot of questions. The first one, when troubleshooting milk fat depression issue, how much time do we have to look back to check for a change in the diet or management?
You're thinking how fast does milk fat depression occur if you make some change?
Yes.
So because you're dealing with rumen fermentation, you have a few days involved there. So Kevin Harvatine's group at Penn State have done some studies working out the timing, but as I recall, basically it's around a week for the situation to fully develop, and then it's an equal amount of time to overcome when you make a correction. So that means when you do make corrections, you're going to have to give it a period of time to know if your correction was a good one.
Okay, perfect. I have a second question from William. He's from Peru. Would it be helpful to infuse butyric acid in rumen to avoid milk fat depression?
Interesting question. No, that would not help. You're thinking infuse. I think the question is probably based on that idea. If you infuse vicinic acid, trans 11, then you would not necessarily make the 10-12. But I don't think that would be true.
Okay, here is a question of Matias from Argentina. Can you make some comments about milk fat depression and effective NDF, starch, or NFC?
Well, those are all important. Important in maintaining normal rumen fermentation and then avoiding a situation where you get this shift toward microbial population that would result in this alternative pathways of biohydrogenation. So I think as the questioner would be aware, that's one of those situations you have a lot of interactions going on, but if you are having a problem, that would be a place to look to well, if I was troubleshooting, I would first make sure I really do have a milk fat depression problem, and if I was able to do so, I would run milk fatty acid composition and just make sure I have the trans 10 change so that I'm really dealing with the classical milk fat depression, and then work from there toward changing the effective fiber or the feeding strategy or any of these type of situations that increase the forage, NDF, and effective fiber, these type of situations. I presume there would be some change in the diet that would have occurred or in the feed type that would have occurred that perhaps could help you pinpoint where to focus.
Okay, regarding this, I have two questions. One is, which is the level of milk fat when you say, okay, we have to do something?
Oh.
And the second, if we have several risk factors for MDF, which one is more important to work on first? I mean, which factor has major effects on milk fat depression? Is it fiber or oils? And okay.
That's a hard question to answer. Which because the one that would be most important might differ for each individual situation. I would look, if you have milk fat depression, I would first look at your fatty acids compositions. What are you feeding? Are you feeding dietary supplements, lipid supplements that could be the problem, or some dietary ingredient like the distillers I mentioned that could be the problem? I would look then at, or at the same time, I would look at your diet to think in terms of the effective fiber and NDF. And of course, oftentimes it's easy to change feeding strategies if that has gone through changes that would make it so your feed is tending to be more slug fed than what it was before. So there's not one thing. I think it would depend on your situation, but those 4 or 5 would represent areas that you would very quickly look at and try to assess which ones are more likely to have caused your problem or which ones you can more easily change.
Okay. So I'm going to go with a question from Bill Top. He said feeding palm fat is very widespread. What effect does it have on the trans-10 cis-12 balance? How is it affecting the omega-3 and omega-6 balance in the dairy cow? And will it affect the reproductive efficiency in the Holstein cow?
Oh, Lord.
There's a lot wrapped in that one.
There is, and there's such good work coming out on palm fat, but I'm not involved in any of that work anymore.
Who is doing that?
Adam Locke would be one.
Okay. And
palm fat is not, the C16 is not going to cause milk fat depression. That's one of the advantages of that.
So I guess I might just send that off to Adam.
Sorry.
I'm going up to a question from Salvador Ordaz, and he says he's working in a farm in northern Mexico. His question is, how can he use the RUFL or other variables reported in the forage analysis, and then later he says TMR analysis perhaps, to take better decisions on our farms in terms of the use of bypass fats?
Well, I think, again, I'm not in the field practicing these things, but essentially, I think you have a list of factors that are associated with milk fat depression, risk factors you might call them, and so you would work with this list as you're formulating the diet and try to keep this in mind. I know more and more lipid supplements are added to be able to formulate higher energy diets. These are a challenge, particularly if there's C18-2. That the calcium salts of polyunsaturated fatty acids were protected from becoming available in the rumen. We know now that's not true. They're not inert. They're not protected, but it does offer. It does slow down the rate at which they're disassociated to not adversely affect the microbial population, and also to have less effect on the biohydrogenation steps. So I guess I'm not able to say A, B, C, but rather to say you look at this list of risk factors and then work with that in formulating a diet.
It sounds like we could put together a nice article and put it up on our blog post for some of these questions. I have a question from Alberto Navas. He says, thank you very much for sharing your knowledge. And his question is, what level of pH do you expect to start showing milk fat depression?
Well, again, see, that depends on all these interactions. If I go back to the classic experiments that Greenery did, you can have a low rumen pH as long as you have very low polyunsaturated fatty acids in the diet. It's not going to be a problem. On the other hand, if you have a high level of polyunsaturates, even a more modest change in the rumen pH can trigger the situation, so you have a problem. So it's really due to these interactions rather than thinking there's a particular pH that you can target and avoid and not have a complication.
The summary is it's all very complicated.
Complicated in one sense, but it's really the interactions, and we now know the main players in the interaction. You just need to be able to handle all those.
Okay.
Or use those in terms of troubleshooting if you are having problems.
All right, thank you. Let's see. I'll check and see if Paula has more questions. Paula?
Yes,
Okay.
Marianne. I have a lot.
Good.
Of questions.
I'm actually don't have anything in my window right now, so you can go ahead for a while.
Okay. A question from William. If we feed bypass fats, we don't affect rumen, neither liver function. It just goes directly to the mammary gland. Is it true?
If you feed fats that actually are bypassed, some bypass fats don't bypass completely, but then if those fatty acids are absorbed, then the mammary gland will use them quite extensively, yes. And palmitate's the one that's best characterized, and that's about 15 to 20% of what you supply in a supplement. You'll get that increase in milk fat.
Okay. Another question. Could some byproducts like rice hulls or soybean meal increase milk fat depression? Please make some comments if there is any difference between extruded soybean meal or roasted soybean.
That's a good question, and I'm really not knowledgeable enough to answer that. The rice hulls would depend on the extent to which they affect the rumen fermentation, but on the soybeans, it would be a matter of how fast the oil becomes available, how fast the fatty acids in the different forms of the soybean become available in the rumen. And I'm sure that's known, but I don't recall it right now.
Okay, perfect. Another question. This is from Uruguay. Eduardo is asking you to make some comments about milk fat depression during late winter, early spring, under grazing conditions. It's typical in Uruguay that cows are grazing ryegrass, and in late winter they have this problem with milk fat depression.
Yes. So again, that comes back to these interactions between the dietary factors and the fatty acid supply and the rumen fermentation. But I'm really not going to be able to give any better answer.
Okay. This is a question from William. When cows are lactating, is there an insulin insensibilization? I don't know if that word exists of other tissues than mammary gland.
The answer to that is yes. That one I can answer. The way the body is able to redirect nutrient use is oftentimes through how tissues respond to these acute signals like insulin. And in the case of the dairy cow, with the onset of lactation, the while the mammary gland is not insulin responsive, not sensitive to insulin, the other body tissues like muscle or adipose tissue, their insulin sensitivity is reduced. And what that does then is allow more of the glucose to be, so less of the glucose is used by those tissues, and therefore more of the glucose is available to go to the mammary gland. So the way to think of it is that nature has accorded milk synthesis the highest priority, and so it lets the mammary gland make milk, and the other body tissues have to change their quantity and pattern of nutrients they use to allow the mammary gland to get its nutrients it needs. The same thing happens in late pregnancy, where the fetus, the uptake of glucose by the fetus is also not insulin sensitive, and yet the fetus needs tremendous amounts of glucose in the last phases of development. And so the way the body does that is mothers, other tissues of the cow, of the mother, adapt to use less to allow more to be available for the.
Another question, does lipid mobilization during early lactation influence lipid metabolization of those fatty acids that affects negatively milk fat synthesis?
No, not in general. The mobilization, the fatty acids that are in adipose tissue are the longer chain fatty acids. So C16 saturated and the longer chain unsaturated ones, mainly oleic, C18-1 oleic. And so when they're mobilized, they're circulate as free fatty acids, and the mammary gland takes up quite a high percent of those as they go by and puts them right into milk fat.
Okay, perfect. And the last one, besides milk fat content reduction, is this a pathologic condition for the cow? Should we expect lower carbohydrates digestibility and lower efficiency?
That's also a good question. I think I'm aware of herds that have had milk fat depression for years, several years, and not had any adverse effects on the herd, on the health of the herd or the life of the animals. However, you can easily see that some of these conditions that lead to milk fat depression are ones where you're really upsetting the rumen fermentation. And so the extent to which you would get into some of those situations would not be good for the overall health and well-being of the cow. So I guess a little bit of depression is not really I don't think of it as a health problem, but too extensive, if you have very extensive adverse effects on the rumen, that's not going to be good.
Okay, perfect. Okay, Marianne, I do not have any more questions.
Okay, I have a question. Let's see. This is from Mohammed Lafti. Majd, I'm not sure how to say his last name. Thank you for the interesting presentation. How can we manage our ration to have the same predicted milk fat and milk fat as actually is seen in the cows, because it can affect ME and MP balance and dry matter intake prediction. It is very important to have your intended milk fat and the result be the same or at least close. And I think he's speaking specifically of working within the model.
Oh.
And I know that you haven't had much experience doing that.
No.
Right. I think this might be one that I can toss over to Tom or Sam to address.
Sure.
there's a lot of questions that we've had tonight that are very much on the practical side of this whole milk fat depression issue. And you and I talked last week that was something that it would be great to have one of the more cow-centered nutritionists available to help field these questions. And what we'll do is put together, I'll go through all of these questions and see if we can't put together a really good topic of discussion. And as always, when I'm looking for speakers for the nutritionist, I try to keep in mind what people are really, really focused on and trying to solve in their herds and their issues. So it certainly is, I think, unless we get some more questions, I don't believe Paula has any more, and I have no more questions in my window. So we might let you off the hook shortly.
I would agree, Marianne, I would agree with your last statement. I think the progress just in the last few years, progress and changes in lipid supplement, the whole area of lipid supplement, and now this effort to see if you could use fatty acid composition to troubleshoot, or at least a bulk tank sample. Those are all things that are going to make this a lively area here for the next.
Well, and I believe in November, Heather Dan from the Minor Institute is going to be our presenter, and she's been working a lot with Barbano on the questions of changing milk fat and milk fat depression through diet. So everyone that asks questions, stay tuned, I guess. So
excellent.
All right,
thank
Paula.
You.
Thank you so much. This has been a real pleasure. Paula says thank you, and she has no more questions. Thanks very much for joining us. I will close out the webinar, and everybody can digest what they've heard. So,
thank you.
Thanks a lot, Dale.
Good luck.
Thank you. Bye.